Sex determination in Drosophila melanogaster. Drosophila melanogaster is also known as fruit fly commonly, and this mechanism is also known as Genic Balance Theory and Genic Balance in Drosophila melanogaster. So basically, how sex determine in case of Drosophila. Drosophila melanogaster basically contain 4 pairs of chromosomes, and out of these 4 pairs, the last or the 4th pair is sex chromosomes. Male Drosophila commonly carry XY chromosome, female carry 2X chromosomes. So sex determination in Drosophila is not due to the presence of XX and XY chromosomes. As you know, in case of human, XX are the sex chromosome present in female, while XY are those sex chromosome present in male. So basically in human, Y chromosome determines the maleness. The presence of Y basically participating in the development of male. But for Drosophila, it is not the case. So for the Drosophila, Y chromosome is not participating or not playing any role in the formation of male. So these are the chromosomes of Drosophila melanogaster, and you can see here the female Drosophila carry 4 pairs of chromosomes, and the first pair, that is a large size. Similarly, the second pair also large size. So 4 large pair of chromosomes are present, while here is a male and Drosophila, and male Drosophila carry XY chromosome. These are the sex chromosomes along with the 3 pairs of the autosomes. So in the Drosophila, the sex basically dependent upon the ratio of the X chromosome to autosomes. That is known as Genic Balance Theory, and that mechanism is provided by Calvin Bridge in 1921. What are the conclusions or basic points that Bridge conclude? That balance between the ratio of autosomes to X chromosomes determines the sex in Drosophila. कि ड्रोसोफिला मेलानोगास्टर में फ्रूट फ्लाई में जो एक्स क्रोमोसोम्स हैं और ऑटोसोम्स हैं, इनका जो नंबर है वो इम्पोर्टेंट है, और इनकी जो रेशियो आती है वो बेसिकली डिसाइड करती है जेंडर ऑफ द ड्रोसोफिला. One autosomal set in Drosophila is equal to 3 chromosomes. So normal Drosophila carry 2 autosomal sets, means उसमें 6 chromosomes हैं, autosomal chromosomes हैं, और इन 6 autosomal chromosomes के साथ साथ उसमें 2 जोन से हैं वो sex chromosomes हैं. So ये एक table है, and that table is basically supporting the bridge theory of sex determination by genic balance in Drosophila. You can see here the number of the X chromosome, number of the autosomal sets, and then total number of chromosomes present in that Drosophila, then X autosomal ratio, and number of the X chromosome divided by the number of the autosomal sets. उनकी जो भी रेशियो आती है वो यहाँ पे कंक्लूड की गई है. On the basis of this ratio, the gender of the Drosophila will be decided. So if the number of the X chromosomes are 3, number of autosomal sets are 2. 2 autosomal sets means 1 autosomal set is equal to 3 chromosomes. So 2 autosomal sets is equal to 6 chromosomes. Total number of chromosomes are 9. 6 autosomes plus 3 X chromosome. So ratio, number of the X chromosome divided by number of autosomal sets. So X chromosome का number 3 है, उसको आप divide कर दें autosomal set के number से, that is a 2. 3 by 2 is equal to 1.5, and the sex of the Drosophila is meta female. So इस base पे जौन सा है Drosophila में sex decide होती है. Similarly, if number of X chromosomes are 3 and autosomal sets are also 3, the ratio is 1. 3 by 3 is equal to 1, and the sex of the Drosophila is female. Similarly, if 2 X are present along with 2 autosomal sets, the ratio is 1. That will also form the female. When 3 X are present along with 4 autosomal sets, the ratio is 0.75. That will form the intersex. And if 2 X chromosomes are present along with 3 autosomal sets, then the ratio is 0.67. 2 divided by 3. So the answer is intersex. If one X chromosome is present along with two autosomal sets, then the ratio is 0.5 and the sex of the Drosophila is male. Similarly, if XY are present along with two autosomal sets, so one X and two autosomal sets are there, so the answer is 0.5 and sex is male. And if XY is present along with three autosomal sets, the answer is 0.33 and the sex of the Drosophila is meta male. So we can conclude that if X autosomal ratio is equal to 1, it will form the normal female. If X autosomal ratio is equal to 0.5, it will form the normal male. If X autosomal ratio is more than 1, it will form a meta female. Meta female is basically a sterile female, not normal. If X autosomal sets are less than 0.5, it will form the meta male. And if X autosomal ratio is between 0.5 to 1, it will form the intersex. So Bridge basically explained his results that non-disjunction is basically the reason behind the formation of genetically imbalanced gametes. And due to the non-disjunction, two types of eggs are formed by a female: an egg with two X chromosomes or an egg with no X chromosome. So both types of eggs have the normal three autosomes. So here is a female Drosophila. Female Drosophila carry two X chromosome along with two autosomal sets. ठीक है, इस ड्रोसोफिला में अब आपके पास क्या है कि दो एक्स क्रोमोसोम्स हैं और इन दो एक्स क्रोमोसोम्स के साथ साथ इसमें जौन से दो ऑटोसोमल सेट्स हैं जिसमें 6 क्रोमोसोम्स हैं। अब ये ड्रोसोफिला जो है ये मियोसिस में पार्टिसिपेट कर रही है, गैमीट फॉर्मेशन में जा रही है और इसने क्या करना है, इसने मियोसिस 1 हो रही है और मियोसिस 2 हो रही है। मियोसिस 1 जो है नॉर्मली हो रही है, मियोसिस 2 में आप देख रहे हैं मियोसिस 2 में नॉन डिसजंक्शन हुई, एज अ रिजल्ट ऑफ विच one gamete is formed by that female that carry two types of two X chromosomes. So ये एक ऐसा गैमीट बना है जिसमें दो एक्स क्रोमोसोम है, जबकि इसके साथ जो गैमीट बना है दैट इज विदाउट एक्स क्रोमोसोम। अब ये दो एक्स क्रोमोसोम्स के साथ साथ इसमें जौन से हैं आपके पास ऑटोसोम्स का नंबर नॉर्मल है गैमीट में, मतलब 3 ऑटोसोम्स हैं। और ये जो गैमीट आपके पास आ रहा है जिसमें कोई भी एक्स प्रेजेंट नहीं है, उसमें भी आपके पास 3 ऑटोसोमल जौन से क्रोमोसोम्स प्रेजेंट है। अब क्या होगा कि अगर आपके पास ये एक गैमीट है दैट कैरी 2 कॉपीज ऑफ एक्स क्रोमोसोम्स, that is a diplogamete, and if diplogamete is fertilized by normal haplogamete, it will form the ट्रिप्लॉइड केस। या फिर आपके पास क्या आ जाएगा कि अगर यहाँ पे दो एक्स क्रोमोसोम्स हैं इस गैमीट के अंदर और अगर इसको कोई नॉर्मल गैमीट फर्टिलाइज करेगा तो इसके रिजल्ट में जो भी ऑफस्प्रिंग बन रहा है दैट कैरी 3 कॉपीज ऑफ एक्स क्रोमोसोम। सो double X egg fertilized by a Y-bearing sperm, the result is XXY fly. इस गैमीट की बात हो रही है कि ये जो एग बना है इसमें डबल एक्स की कंडीशन है, ठीक है। अगर इसको वाई बेरिंग स्पर्म फर्टिलाइज करेगा तो एज अ रिजल्ट ऑफ इट XXY fly is there and that is a normal fertile female। एंड इस पे फोकस करें कि ये फीमेल है, although Y क्रोमोसोम इज प्रेजेंट हेयर। ह्यूमन में हम ये पढ़ते हैं कि ह्यूमन में अगर वाई क्रोमोसोम प्रेजेंट है तो वो मेल्स बनते हैं। आपको याद है क्लाइनफेल्टर सिंड्रोम, लास्ट टाइम जॉन सा मैंने अपने पिछले लेक्चर में जॉन सा है वो डिस्कस किया था कि क्लाइनफेल्टर सिंड्रोम में आपके पास जो कैरियोटाइप होती है दैट इज डबल एक्स वाई and Klinefelter syndrome is present in male because Y chromosome is present. But in case of Drosophila, Y chromosome did not play any role in the maleness. That's why डबल एक्स वाई फ्लाई जो है वो आपके पास नॉर्मल फर्टाइल फीमेल है। इफ द सेम डबल एक्स एग जो पीछे जोन से आया है दैट इज फर्टिलाइज्ड बाय एक्स बेरिंग स्पर्म, इट रिजल्ट्स इन ट्रिपल एक्स एम्ब्रियो। There is a viable sterile fly designated as super female or more appropriately meta female. Meta female, previously known as super female, but later on it was named as meta female. So if diplo egg, double X egg is fertilized by X pairing sperm, it will result in the formation of triple X embryo. Egg without an X chromosome fertilized by a Y bearing sperm. पीछे आपके पास दो तरह के एग बन रहे थे, एक में दो एक्स क्रोमोसोम पास हुए हैं और दूसरे एग के अंदर कोई भी एक्स क्रोमोसोम प्रेजेंट नहीं है. सो इसकी बात हो रही है कि एग विदाउट एन एक्स क्रोमोसोम फर्टिलाइज्ड बाय अ वाई बेरिंग स्पर्म रिजल्ट्स इन YO एम्ब्रियो व्हिच डज नॉट डेवलप. सो YO एम्ब्रियो जो है ये सरवाइव नहीं करती. Why? Because X chromosome is basically necessary for the survival. The genes of the X chromosome are basically participating in the survival, or they are forming an essential product. That's why YO embryo does not develop. Similarly, an egg without an X chromosome fertilized by X pairing sperm, it results in XO sex chromosome constitution and phenotypically normal, but these are sterile male. So X0, basically in case of human, that is a Turner syndrome that affects the female, while in case of Drosophila, X0 is the sterile male. Why these are sterile? Because Y chromosome is absent, and it is indicating that Y chromosome, although do not play any role in the development of male, but they do play an important role in the male fertility. Y chromosome मेल ड्रोसोफिला डेवलपमेंट में पार्टिसिपेट नहीं करते हैं, लेकिन Y क्रोमोसोम मेल फर्टिलिटी के लिए नेसेसरी है. So Y chromosome does not determine maleness. At least one X must be present to produce a viable embryo. जैसे हमने पीछे देखा कि YO एम्ब्रियो, Y0 एम्ब्रियो जब आपके पास आती है, X क्रोमोसोम उसमें एब्सेंट है, तो वो एम्ब्रियो सर्वाइव नहीं करती. So it means at least one X must be present to produce a viable embryo or for the survival of the embryo. Y chromosome is necessary for the male fertility. And triple X constitution is an abnormal female phenotype, not always viable and termed super female or meta female. So these are the conclusions of the bridge experiments. Double XY individual were normal fertile female, X double Y were fertile males. Ratio of X chromosome to autosome is important for sex determination. So ये basic conclusion थी ब्रिज के एक्सपेरिमेंट की कि जो ड्रोसोफिला है उसमें सेक्स डिटर्मिनेशन डिपेंड करती है ऑन द रेशियो ऑफ एक्स क्रोमोसोम टू द नंबर ऑफ ऑटोसोमल सेट्स. So what is balanced theory of sex determination? That is restricted to Drosophila melanogaster. So ये केस जस्ट हम ड्रोसोफिला पे अप्लाई करते हैं, ये ह्यूमन पे अप्लाई नहीं होता. The X chromosome contain genes that control femaleness. The autosomes trigger the expression of male determining genes. The ratio between the number of X chromosome and number of complete set of autosomes determines the sexual phenotype. So ये points क्या हमें बता रहे हैं कि जो X chromosome है, basically it carry the genes that is necessary for the female development. Autosomes or the autosomal genes are necessary for the expression of the male determining genes in case of Drosophila. And the ratio between the X chromosome and the number of complete set of autosomes decide the sexual phenotype of Drosophila. When single X chromosome is present along with two set of autosomes, it will form a male. So 1X along with 2. So 1 by 2, number of X chromosome divided by number of autosomal sets, the answer is 0.5. And we have discussed previously that if ratio is 0.5, it will form the male. The addition of another X chromosome with two set of autosomes form female. So if two X chromosomes are present along with two autosomal sets, the answer is 1. And if the answer is 1, it will form the female. So which basic genes are participating in the sex determination of Drosophila? The first gene is named as transformer gene, TRA, that is an autosomal recessive gene impact on the sex determination of Drosophila. If female are homozygous for transformer, they are transformed to sterile males. while if male are homozygous for transformer, they are unaffected. So transformer gene basically affected the female Drosophila. Another gene is sex lethal gene that is located on the X chromosome. If sex lethal gene is on, it is participating in the development of female. If sex lethal gene is off, inactive, it is participating in the development of male. So sex lethal gene is basically a master switch gene in sex determination of Drosophila. If X autosomal ratio is equal to 1, it will activate the sex lethal gene and development of female start. If X autosomal ratio is equal to 0.5, it will deactivate the sex lethal gene and development of male will start. If mutation takes place in sex lethal gene, it will kill the female embryo but has no effect on male embryo. That's why it is known as sex lethal, because its mutation can kill the female embryo. Why it is not affecting the male embryo? Because in males, sex lethal gene is off. Sex lethal transformer, and the third gene is the double sex gene. These three genes are most important regarding the sex determination of Drosophila. Sex lethal gene, that is a master switch gene, and further regulates the expression of transformer and double sex gene. When sex lethal gene is activated, it further activates the wild type transformer only in females. Transformer further controls the expression of double sex. Double sex protein has ability to activate genes required for either male or female sexual differentiation. Double sex gene is expressing both in male and female, but its expression or its product is different for the male and for the female. What are the numerator elements and denominator elements? Numerator elements include the genes present on the X chromosome that regulate the sex lethal gene into on state. न्यूमरेटर एलिमेंट में वो जीन आते हैं जो एक्स क्रोमोसोम पे प्रेजेंट हैं और वो सेक्स लीथल को रेगुलेट करते हैं और उसको बेसिकली एक्टिवेटेड फॉर्म में रखते हैं. और ये अगर सेक्स लीथल जीन ऑन स्टेट में है, एक्टिव है, तो इट विल पार्टिसिपेट इन द डेवलपमेंट ऑफ फीमेल एंड इट basically depend upon genic balance of X and autosomes. While denominator elements are the genes present on autosomes that regulate the sex lethal into off state and it further participating in the development of male. So दो तरह के आपके पास जीन्स हैं. वो जीन्स जो एक्स क्रोमोसोम पे हैं और सेक्स लीथल को एक्टिव रखते हैं वो न्यूमरेटर एलिमेंट्स हैं, जबकि वो जीन्स जो ऑटोसोम्स पे हैं और सेक्स लीथल को ऑफ कर देते हैं वो डिनोमिनेटर एलिमेंट्स हैं. सो न्यूमरेटर एलिमेंट्स फीमेल डेवलपमेंट में पार्टिसिपेट कर रहे हैं, जबकि डिनोमिनेटर एलिमेंट्स मेल डेवलपमेंट में पार्टिसिपेट करते हैं. जैसे पीछे भी जिक्र हुआ था कि ऑटोसोम्स बेसिकली ऑटोसोम्स पे वो जीन्स हैं जो मेल डेवलपमेंट में कंट्रीब्यूट कर रहे हैं. सो हाउ सेक्स लीथल is acting as a master switch gene. Basically, sex lethal gene counts the number of X chromosomes. If two X chromosomes are present, sex lethal become activated. Like in case of female, when two X chromosomes are present, sex lethal activated. How it counts the X chromosome? It depends on the protein product of numerator elements. Numerator elements are those genes present on the X chromosome. So अगर वो जीन जो है वो ज्यादा प्रोडक्ट बना रहे हैं तो वो प्रोडक्ट जो है उसके एक्सप्रेशन को देखते हुए सेक्स लीथल जीन एक्टिवेट होगा. If the level of protein of numerator elements is high, sex lethal gene switch on. and participating in the female development. But if the level of the protein of numerator elements is low, जैसे मेल है, मेल में सिंगल एक्स क्रोमोसोम है, तो उसके सिंगल एक्स पे जो भी प्रोटीन्स हैं वो ऑब्वियसली कम अमाउंट में बनेंगी। तो उस टाइम पे सेक्स लीथल जीन जब ये देखेगा कि वहां पे जो प्रोटीन बन रही है उसका लेवल लो है, तो वो स्विच ऑफ हो जाएगा। और स्विच ऑफ हो, अगर सेक्स लीथल जीन स्विच ऑफ होगा तो इट विल फॉर्म द मेल्स। So that's all about the sex determination in Drosophila melanogaster, or the sex determination or the genic balance theory of fruit fly.